This is the third video for the cellular respiration unit. In this video, we will be discussing the final stage of cellular respiration, the electron transport chain, and its role in oxidative phosphorylation. The electron transport chain is located in the inner mitochondrial membrane. In this figure, the intermembrane space is shown below the membrane, so the outer membrane of the mitochondria would be down here. There are 4 different protein complexes that make up the electron transport chain, labeled complex 1, 2, 3, and 4. These complexes have 2 roles. They bind and transfer electrons, and complex 1, 3, and 4 also pump protons from the matrix into the intermembrane space. They use the energy from the electrons that they are transferring to power the proton transport. Thus, this is active transport and is creating a concentration gradient of hydrogen ions with a high concentration in the intermembrane space and a low concentration in the mitochondrial matrix. The 5th protein found in the membrane is called ATP synthase, and this enzyme is responsible for producing ATP through oxidative phosphorylation, using the energy found in the electrochemical gradient of hydrogen ions. Those ions flow down their concentration gradient through ATP synthase, providing the energy to produce ATP. This will be shown in the following slides. The first step of the electron transport chain is that the electrons from the first 3 stages are brought to the electron transport chain through the electron carriers. NADH drops its electrons off at complex 1, being reduced back to NAD+, while FADH2 drops its electrons off at complex 2, being reduced back to FAD. The NAD and FAD are now free to go back to the first 3 stages to pick up more electrons. Once bound to the complex, the second step is that the electrons are then passed from complex to complex down the electron transport chain. As they are passed, the energy in those electrons is used to pump hydrogen ions from inside the matrix out to the intermembrane space. This produces an electrochemical gradient of protons that then flow back through ATP synthase, providing the energy to produce ATP from ADP and inorganic phosphate. This is the step 4 of this reaction. ATP synthase is able to couple the flow of protons to the generation of ATP because it has 2 distinct subunits, F0 and F1. When protons flow through the F0 channel in the inner mitochondrial membrane, it causes the channel to rotate, similar to water in a flowing river passing through a water wheel. This converts the energy of the proton gradient into mechanical rotational energy, a form of kinetic energy. This mechanical energy is transferred to rotation of the F1 subunit, which allows it to catalyze the synthesis of ATP from ADP and inorganic phosphate. That process is called oxidative phosphorylation. The final step of the process involves removing the electron from the complex 4 of the electron transport chain. Each protein complex can only hold a set number of electrons. Thus, if the electron is not removed from complex 4, then electrons from complex 3 will no longer be passed to complex 4. If they are stuck in 3, those from 2 will no longer be passed to complex 3, and so on and so forth until the entire chain will stop flowing. Without electron flow, there is no energy to pump the hydrogen ions, and without the concentration gradient, no ATP can be made. Thus, it is very important to remove the electron from the end of the electron transport chain. The molecule that does this is oxygen. oxygen functions as the terminal electron acceptor and is reduced to water as it takes electrons from complex 4. In fact, this is the only reason you need oxygen to live. Without it, the electron transport chain cannot run, and you cannot produce enough ATP to power the cellular processes required for life. To calculate the total amount of energy produced from ATP, we need to compile all 4 stages. In the last video, we showed that stages 1 through 3 produced a net yield of 4 ATP, 10 NADH, and 2 FADH2. Each of the NADH drops its electrons off at complex 1, which produces enough proton gradient to generate 2.5 molecules of ATP. Thus, for every 1 NADH, we produce 2.5 molecules of ATP. Because FADH2 drops its electrons off at complex 2, it does not generate as much of a concentration gradient. So each FADH2 is equivalent to 1.5 ATP molecules. Using this information, we can determine that 10 NADH will produce 25 ATP through the electron transport chain, while 2 FADH2 will produce 3 ATP through the electron transport chain. That produces a total of 28 molecules of ATP through oxidative phosphorylation, combined with the 4 through substrate level, gives us a total of 32 ATP for all of cellular respiration.